Okay, so it's a great pleasure for me to introduce Dr. Alwan. She has a PhD from Ohio State University. She worked with Manoj Srinivasan. Now she's currently postdoc in the College of Medicine in the Department of Physical Medicine Rehabilitation. So, well, we been very interested in her work on human locomotion and modeling of complex movement, and so we thought was a great fit for our program, and we are very interested to learn and to hear from her about her research. So without stealing her too much time, let's thank for Dr. Alwan for a kind availability of being here with us.
Yeah, thank you, Alessandro, for the introduction and for the invitation. Today I'll be presenting work from my PhD on addressing complexity in biomechanics, focusing on human locomotion models, analyses, and experiments in complex movement tasks. To understand and analyze human movement, we use one or a combination of the following quantitative measures to describe locomotive phenomena. Kinematics describe the motion itself, things like positions, velocities, and accelerations, tells us what the movement looks like. Kinetics studies the forces that cause the motion, such as ground reaction forces or joint torques. These help us understand why the motion occurs. Third, energy expenditure gives us a measure of how much energy we're spending or efforts required for certain movement tasks. And finally, we may consider other clinical and physiological metrics that can inform rehabilitation, performance, or understanding movement disorders. These measures give us a comprehensive understanding of how and why humans and animals move the way they do. And I'm especially interested in human-legged locomotion, including gait. So our body dynamics, like the muscles and the joints, are guided by signals from sensory systems. These signals are processed together to figure out the current state of the system and compare it to a target state or equilibrium, like reaching a certain target or staying balanced and upright. The controller then adjusts our movements by sending the signals back to the body, creating the joint torques or muscle excitations to keep us steady. Simple models have proven to be effective in predicting movement. We use simple models to help explain complex motor behavior using data-driven mathematical modeling and provide accurate predictions of legged locomotion drawn from experimental data sets. In this example here, the researchers have added a mass to one of the feet, and they used a simple point mass model to see how the step length asymmetry and step time asymmetry transients, they respond to such adaptation. So in the beginning of the trial, they have the people walk normally, and then they add the foot mass. This is a shaded blue area. That's called the adaptation period. And then there's the de-adaptation, where they remove that foot mass and see how people react. And they use a predictive model based on only energy and symmetry. They found that the energy and plus the symmetry cost term represents better the experimental data, as you see here through the dark blue line. So the step length asymmetry is 0 when they're normally walking, and then during adaptation period, it goes to negative. And that's like the fast transient period, and then slow transient period where it comes back to steady state. And then when they remove the foot mass, it kind of shoots a little bit more to the positive, where the brain thinks that the foot mass is still on, but so you might take like longer step length even though you don't need to. And then there's this includes the de-adaptation period. So the idea here is the model predicted well the experimental data, and this is one example of many examples where we use simple models to understand complex behavior in locomotion, or help us answer questions in biomechanics. So my talk today will involve 3 studies from my PhD research, and it focuses on addressing current challenges in the biomechanics field. So one of the challenges that researchers see is the misestimation of muscle metabolic cost using modeling and simulations. Our study proposes a new averaging method of muscle metabolic cost to account for stride-to-stride variability. Another challenge is where biomechanics studies often involve zero slopes, and the second study develops predictive models based on experiments to understand the control strategies while walking up and down slopes. The third challenge is having short trial durations or limited data, and our third study implements a novel application of a statistical estimate to enhance estimation accuracy in biomechanical analyses. So diving into the first study, a little bit of background. Human walking is not exactly periodic and contains intrinsic stride-to-stride variability. Possible sources of this variability could come from sensory noise or motor noise. Sensory noise is the uncertainty in state estimation. This results from loss of sensory precision, making it harder for the brain to know exactly where the body is in space. For motor noise, this is signal-dependent fluctuation in the motor commands, and this comes from involuntary variations that exist in the force the muscles actually produce. Another source of variability is intentional or exploratory variability. This comes from the nervous system to promote motor learning. For example, if you have a mass added to your foot or you're trying to learn a new task, your body might actually want to search for the most efficient movement pattern by exploring different possible solutions. So in this example here, this is one participant walking at different speeds, and each dot represents a different step. So for example, if we're looking at 1 meters per second speed, the different dots represent different steps. And this shows like the variation of the position of one of the feet, which shows variability and from stride to stride. And the body uses corrective feedback control strategies to maintain stable walking and not fall down. Those can include like foot placement control, push-off modulation, ankle and hip strategies, and other strategies that might include like increasing step width or cadence. And the one that we're going to be focusing today in generally is the foot placement controlled method. And there's substantial evidence that humans optimize energy. It's the energy optimality criterion. Humans minimize their metabolic costs at roughly 1.2 meters per second for healthy adults. And the idea here is that the energy optimality theorem is widely used to understand certain locomotive phenomena. Energy cost is likely affected by stride-to-stride variability. It has been shown that increases in step length or step width variability may be correlated with measured metabolic cost, and that stabilizing feedback control actions may contribute to increased energetic demands. Typically, total metabolic costs of walking can be estimated using muscle energy models, using simulations, or indirect calorimetry, where we ask people to wear masks and we measure their O2 input, CO2 output, and we use an empirical formula to compute the total energy rate. Muscle energy models specifically are helpful for targeted energy estimation of specific muscles or muscle groups, especially for those with pathological gait, to understand why there's some issues with walking, with their gait, or obtaining metabolic costs for scenarios difficult to test in a lab setting. Some of the example muscle energy models that are used is the Emberger and Bergava models, with a general form of the total energy rate is a sum of the work rate and a bunch of heat rate terms. And OpenSim, which is a musculoskeletal simulation software, implements muscle energy models to estimate a theoretical metabolic cost. So researchers often estimate metabolic costs using averaged information from a single random gait cycle or multiple gait cycles. What we mean by here is the first method here that researchers often use is just to use a random stride, and that obviously does not include any stride-to-stride information, so that could misestimate metabolic cost. The other method that researchers do is they use the averaged motion and force information or gait information from multiple strides, and then use that as input to compute metabolic cost. These methods misestimate cost due to ignoring stride-to-stride variability. The objective here is the objectives of this study is to evaluate metabolic cost errors qualitatively, and also estimate the cost of variability due to motor sensory noise using modeling techniques. The second method that I was talking about earlier can be explained by Jensen's inequality. So our hypothesis is that the average gait patterns will have lower metabolic costs compared to the multi-stride averages. Because metabolic cost functions are convex in theory, the general form of Jensen's inequality states that the function evaluated at the average value of 2 points is always less than or equal to the average value of the function evaluated at those 2 points. And I'll explain that more in the next slide as well. Next slides, I guess. So we use publicly available data set that includes motion and ground reaction force data. We use 8 able-bodied subjects, 5 of which are female, 3 are male, and 3 of which are female. We randomly selected 5 consecutive strides from 1 minute of normal walking at 1.2 meters per second speed. So we used the walking data as input to the OpenSim software. For simplicity, we use the Gait 2354 model, which has 23 degrees of freedom and 54 muscle tendon actuators. We scaled the human body model to match the anthropometric properties of each participant. And we use that information as input for the inverse kinematics tool, which estimates joint angles. And from there, we computed the muscle states that includes activations using the computed muscle control step. And from there, we estimated metabolic costs using the Emberger model probe that is available in OpenSim. We also demonstrate the misestimation of metabolic cost in a similar manner using a torque-based effort estimate, but for the sake of time, we will skip that today. So again, people either run the simulation for one random stride, or they average stride information from multiple. What this looks like is, imagine you have ankle angle, for example, for 5 different strides, and people average that information to have one representative average stride. And what we are proposing is to average the metabolic cost for each stride separately by running each stride separately once, getting a metabolic cost of that stride, and then do the stride 2, 3, 4, and the next, and then take the arithmetic average of all of these metabolic costs. And that's what we're calling the gold standard, the one that would fully take into account the stride-to-stride variability. Additionally, we use an inverted pendulum model, which is a simple simple walking model that is widely used in dynamic walking principles to illustrate the cost increase due to stride-to-stride variability. The inverted pendulum model has step-to-step transitions involving impulsive push-offs and heel strikes. And we model the sensory and motor noise as applied to forward velocity that results in stepping variability. We also use a feedback controller. So the sensory input is the feedback controller. The sensory input to the feedback control is the forward speed, and the sensory noise is implemented as Gaussian added to this forward speed, which results in stepping variability. We also use nominal ideal gait to be energy optimal at a given speed. The deviations then are corrected using a feedback controller that adjusts foot placement and push-off. We model metabolic cost as the energy cost from work done by push-off impulse and the leg swing cost.
Just to make sure I'm understanding, you have the inverted pendulum model, and then you add additively the noise, correct?
Yes.
Okay, good.
We found that the costs have stride-to-stride variability, and the average gait pattern has lower costs. So method 1 is the cost of individual strides. Method 2 is the cost of the average gait pattern. Method 3 is what we're proposing, the cost averaged over multiple strides. And here the dots, each color is one participant's data, and each dot is a different stride. So the cost of individual strides vary, and as you can see here, when compared to the cost averaged over multiple strides from method 3, you could either underestimate or overestimate. So anything below that line is underestimation, above that line is overestimation. Here we're representing the metabolic cost estimation error, always according to what we're proposing as the gold standard, method 3. The diamond scatter points here are the error due to the cost of averaged gait pattern. And as you can see, which we explained by Jensen's inequality earlier, is that you're essentially removing the variability and you're underestimating metabolic cost consistently. So the error due to randomly chosen stride can go up to 10% of underestimation or overestimation, but the average stride from multiple can go up to 3% of underestimation. For the simple biped model, it predicted the increase in metabolic cost. We used the effect of motor noise, and we found that the mean cost and the standard deviation of the cost from stride to stride increase as the noise and motor actions increase, as well as in sensory noise. We see a similar trend where the standard deviation, the mean cost also increase as you increase the sensory noise. So we've seen that neglecting stride-to-stride variability in metabolic cost simulations may misestimate the cost up to 10%. The simple inverted pendulum model shows that the sensory and motor noise increases metabolic cost, and these insights advance our understanding of metabolic cost modeling and provide a foundation for refining predictive methods in human energetics within biomechanics. We could repeat this study in overground walking conditions to simulate natural human walking, or to include participants with movement disorders to see how amplified the errors are. We could also simulate stepping variability by adding motor and sensory noise simultaneously rather than separately. Now on to the second study. Falls are the second leading cause of unintentional injury deaths worldwide, and over half of the reported fall injuries occur during walking on slopes and stairs. It is important to focus on fall risk prediction because it can inform meaningful intervention, especially in clinical contexts. Slope walking is more challenging for healthy people than level walking, and it's even more challenging for people with mobility impairments. This is due to higher energy demands due to altered muscle activation, inter-joint coordination, joint mechanics, and possible changes in motor control strategies from level walking, which impose balance and stability challenges. As we've discussed before, walking is not exactly periodic, has inherent fluctuations, and people use corrective feedback strategies like foot placement control, which can be used as a predictor of falls. So the objectives of this study is to investigate how the approximate center of mass deviations are corrected using foot placement control, and to assess the stability during up and down slope walking. So stability is roughly defined as not to fall down despite small perturbations. So imagine a curve that sits in n dimensions. This is the periodic orbit in red here. This may represent like one cycle or one step. And the perturbation, imagine the perturbation starting from a neighboring point from the periodic motion and ending also at a neighboring point. And this is a transient non-periodic trajectory. We want to see if the system gets closer or diverges away from the red periodic orbit. Briefly here, we use something called flow K multipliers to quantify stability. So if it's less than 1, the system is asymptotically stable. If it's greater than 1, the system is unstable. And perhaps that could be represented by like a fall. And even if it's less than 1, sometimes we can get a trend on if the system is more susceptible to falls. Like if the human being is more susceptible to fall, or the system is becoming more unstable and getting closer to 1. The idea with this is what we call point crane map, and typically we look at like one system once per cycle, at the system once per cycle, and this is what's called state-based point crane map. We use data from 10 able-bodied subjects at 1 minute of normal walking trials, walking at 1.2 meters per second. We have 9 inclines and declines from -10 degrees to +10 degrees at 2.5 increments. Kinematic motion data was available. From that, we represented the center of mass as the average between the right and left ASIS markers, and to represent the feet, we use the heel markers. So previously, Wang and Srinivasan, they found that we can accurately predict the placement of the next to stance foot from the current center of mass state. And for example, if our inputs are the deviations in the sideways pelvis position and the deviations in the sideways pelvis velocity, we could accurately know how much the foot is placed towards a certain direction. So in this example, a sideways direction. And these linear regression coefficients that we get from this linear relationship is they are called foot placement control gains, and they basically tell us what is the extent of the correction according to a center of mass deviation. So to compute flow K multipliers, we computed a linear model that maps the hip state from left mid stance to the right mid stance, and from the right mid stance to the left mid stance. And by multiplying these individual step maps, it computes the full point crane maps. Again, point crane maps describe the state of the system after one stride or one gait cycle. And we get 3 eigenvalues of the point crane map, and that's what we call flow K multipliers. And we use the 3-state variables, sideways hip position, sideways hip velocity, and fore-aft hip velocity. So these are the foot placement control gains, and I'm only showing illustrative figures for 2 of these gains here, and we've seen that if you look at the decline ones from right to left, it's easier to follow. So as you go steeper in declines, you see that the foot placement modulation decreases, and similarly with going up an incline, the foot placement modulation decreases for the following gains, and we'll explain why that might have happened in next slides. People take wider steps as they go down a decline or down a slope, and then they take wider steps as they go up slopes as well. With step length, it goes down as you go down slopes, and step length went up as you go up slopes. Flow K multiplier, this one corresponds to the perturbation direction with the slowest decay, and it decreases in both declines. So as you go from 0 to -10, flow K multiplier decreases. As you go from 0 to 10, also decreases. And this suggests that humans might be recovering faster from perturbations at steeper slopes. And the other flow K multipliers that represent more stable directions, they remain unchanged with slope. So I'm only showing the one with the significant changes or significant p-values. So we have shown how foot placement control strategies adapt to walking on up and down slopes. So we've shown that the hip states successfully predicted the placement of the next stance foot. This is in line with previous work on level ground walking. And we inferred that it may be less necessary for the foot placement control to be continuously adjusted in response to a hip perturbation, as the stability could be promoted by having a larger step width. And our findings may inform diagnoses and rehabilitation of specific gait pathological populations or improve the design of assistive or prosthetic devices. Future work may include populations with mobility impairments, exploring a wider range of inclines and declines, whether experimentally or using by building it like a feedback controller to simulate the walking. We could also prescribe step width and step length while walking down and up slopes to see if that's actually what people use to modulate their body and make corrections. And we could also apply external perturbations while walking on inclines and declines and see how the body reacts. Moving into my third and last study, the measured biomechanical data in human-in-the-loop or human performance applications are often accompanied with sources of error. These errors usually occur from large gaps, limited sample size, such as in the case of vulnerable populations, measurement noise, high human variability, soft tissue artifact. All of these introduce biases leading to poor accuracy overall. The one that we make the case for is having a limited sample size, especially in populations with restricted mobility pose a challenge in biomechanical studies. So it has been argued that the minimum number of steps for accurate step kinematic variability is about 400 steps, which could be hard to achieve for people who can't fully walk for longer periods. For energy expenditure, using indirect calorimetry, usually you need 4 minutes of walking for the transient state, and then 2 to 3 minutes to get the steady state value for metabolic cost. So that's about 6 to 7 minutes at least of walking to get accurate measurements. So we're introducing the James Stein estimator it's a statistical estimate that estimates the mean of multivariate normal distribution while reducing the variance. It is often compared to the maximum likelihood estimator. Maximum likelihood estimator, computes the parameter values that makes the observed data have the maximum likelihood. So you can think of like the mean, the arithmetic average as one example, the linear regression or nonlinear regression coefficients as other examples. Potential applications is biomedical sciences and sports statistics, the ones that have high variability and small sample sizes. So in the very first paper of introducing JSE, they give you a baseball example, and they consider the truth to be batting averages from a full season, while they estimate MLE and JSE using only partial data set from the full season, which is 45 at-bats. And the idea is that you compute JSE, and the JSE has smaller error than the MLE in terms of some squared error. So what about like applying the JSE to biomechanical data sets? Imagine having several participants or subjects data sets. What one might do is compute the MLE and then compute the JSE and try to compare the error when you're try to compare the error to a true value. Usually the truth is not known. We can try to approximate it. In terms of this study, we assume that we know we're approximating the truth because we have a full data set. For demonstration purposes, we are estimating the true value. So the idea is that the JSE will have lower error compared to the MLE or simple averages by some rate. So the objective is to apply the JSE to biomechanical estimation problems to improve the parameter estimation accuracy, especially when the sample size is limited. Because when you have people walking at 25 steps versus 200 steps, your accuracy might be compromised, and the JSE is supposed to enhance the accuracy. So we applied the JSE to 3 parameter estimation problems, not to dive too much into the details of these, but we've seen this before in the previous in a previous slide. The foot placement gain, we've used just one gain and apply JSE on. The second estimation problem is a energy rate of people walking around in circles. And the third estimation problem is a resting energy rate. And we estimate the MLE based on the coefficient from linear regression for the first estimation problem. The second is the steady state value from the exponential fitting for metabolic cost. And the third is simply the arithmetic average for the resting energy rate. We approximate the truth to be the full data set, whatever data set that we had collected, which is 100 steps here, 54 data points here, and 40 data points in the last one. And the MLE or the JSE, the sample size for them was, we tested multiple, and I will be only showing you like one slice of that. So for here, 25 steps, 21 data points, and 20 data points for the last one. So the way that the JSE is computed is that for each individual participant, you estimate the JSE by using the global or grand average of the sample data of all participants. And that's what we call the MLE grand average, plus some shrinkage factor times the difference of the individual MLE estimate minus the MLE global average. And so the idea is that the JSE would shrink down or up some values to get closer to the truth. So one important note here is that the JSE does not use any information from the truth data set. It simply uses the information from the data that you already have, which could be the arithmetic average of some data set that you have. And we estimated the sum squared error for the MLE versus the JSE for the purposes of this study to show that the sum squared error for the JSE is actually lower. And we're always using the truth as baseline. So the JSE shrinks data towards the MLE global average, as we're seeing here. One important note is that the JSE has the same grand average as the MLE, so it doesn't change the central tendency, but it does move the individual points more towards the truth and hence decreases variance. And so the truth global average here is in dashed green. The MLE global average here is in dashed blue. And each of the individual points is a different participant. And what we're seeing here is that the JSE is being pulled more towards the truth ground average for all of the estimation problems, remarkably without knowing anything about the truth. And see here, the idea is that the JSE, as you have more variance in the data, it shrinks the data even more. So this means the 0.8 value of C means there's almost 18% shrinkage that have occurred. And the higher the C, the less shrinkage that have occurred. And this makes sense because with sitting metabolics, for example, variance is not high. There's not much variability in the data, but then if you're looking at foot placement control, you can imagine there's more variability in the data, and that's why JSE might be more applicable here, or makes more sense to be applied in these estimation problems. So we only looked at one slice of what we've done. We used 25 numbers of steps for foot placement control, 21 for walking metabolics, and 20 for sitting metabolics is what I've shown earlier. But we wanted to look at the sum squared error if we use very low number of steps versus very high number of steps, as we're getting more closer to the sample size of the truth data. And we've seen that the sum squared error, the JSE actually reduces error for a range of data sets, moving the estimates closer to the truth, and it reduces the relative error even more for shorter data sets. So for sitting metabolics, for example, you see that the sum squared error or the relative error is not quite high, which might mean that JSE might not be needed in this situation. JSE demonstrated its potential to enhance parameter estimation in biomechanical studies, especially when working with smaller sample sizes and larger variability. It reduces sum squared error by using population level information and shrinking the values toward the true estimate. Again, remarkably, without prior knowledge of this true value. Any of the true information that I input on these slides, it's for demonstration purposes to show that JSE actually works. And the JSE could enhance prediction accuracy in healthcare applications where limited sample size exists. Future work could consider biomechanical data of populations with limited mobility, those that are short and variable, and JSE can be applied to biomechanical estimation problems or in the context of a human-in-the-loop optimization, for example, when a large number of trials may need to be performed. All right, moving on to my current work as a postdoc in clinical biomechanics. Here I move more into applied contexts in biomechanics that is very interesting at the intersection of arts and dance. I'm advised by Dr. Lise Worthen Chaudhary in a multi-site study by Ohio State and Yale. So we use participants that are breast cancer patients diagnosed with chemotherapy-induced neuropathy who are 40 plus. And the objective of the study that I'm currently collecting data and analyzing data for is to investigate how CIN affects neuromotor control strategies and how music dance training affects neuromotor rehabilitation by potentially stimulating neuroplasticity. Neuroplasticity broadly is the ability of the nervous system to remodel and rewire in terms of structure, connectivity, and function in response to stimuli that may be external or internal. And dance has the unique ability to motivate physical activity engagement, which stimulate neuroplasticity relevant to rehabilitation and pathological populations. And it has been shown before with Parkinson's disease patients whom their symptoms are even worse than people with CIN. It seemed like there's decrease in freezing of gait, there's more smoothness in walking, there's improvement in some gait metrics that are important, like gait speed, due to dance training or intervention. So the study protocol randomizes participants into standard of care or tango intervention, and we collect a bunch of data, and the one that I'm mostly responsible for analyzing is the gait function to investigate the variability from step to step and the dynamic balance of people walking over ground in a 6-minute walk test while they're instrumented with dual head IMUs that collect AMG and IMU data. And the results from a pilot study on gait variability, we show a trend towards improving key gait metrics like gait speed and reducing the stride to stride variability post-intervention. And that's all I have. Any questions?
So let's thank Dr. Alwan for the great presentation, though you can probably not hear the claps from remote. So I usually risk to monopolize the Q&A session. So I just want to open the stage for possible interactions with the audience.
Yes, I had a
Yeah,
question
go ahead.
about JSE. So if I'm understanding correctly, it's you're introducing bias, so it's no longer an unbiased estimator
Yes.
because you are specifically focused on estimating, like figuring out what the true value is, not just like modeling it, not just maximizing likelihood.
Right. So that is true. That's one limitation of JSE that introduces bias. So at the cost of, or it reduces variance at the cost of introducing bias. And the idea here is that if one researcher wants to use it for their own estimation problem, it might be good to explore a wide range of like C values, shrinkage values, to see what would be an optimal shrinkage value. So we're not introducing too much bias. I know bias is something that statisticians and mathematicians, they don't like to introduce in data analysis. And so this could be one thing that one can do, especially that we have lots of data that exists online and that are publicly available, we can use as testing for our future like research protocols.
Yeah, I mean it makes sense because if you assume that there's some global average that the values probably tend towards, then
Right.
introducing that shrinkage makes sense. I'm curious why JSE? Is it just because it's like a very simple, very interpretable
Yes,
implementation?
it's computationally simple, and it doesn't need a prior knowledge, and so
Yes.
these are the two advantages of using JSE. It's very simple to use computationally as well, which is a huge advantage.
Yes, that makes sense. I hadn't heard of it before. I know that there are other estimators that deal with like bias variance trade-offs, but it makes sense that it's just a very simple interpretable method. So thank you.
Thank you.
Maybe I can ask a question. It's on the first part of your presentation, so part 1 out of 3, about step-to-step variability. So my question is this. It applies to metabolic cost, but it might apply also to other statistic computing on each of the strides. So when you have a time series, in general, you might have a component which is deterministic, a component which is stochastic, and so on. There is one issue, which is the fact that sometimes one of the two confounds the effect of the other. Now, if you have something which is absolutely deterministic, then there is no strict variability in the stochastic sense. Now, let's say that we've taken that into account. So we just talk about the step-to-step variability stochastic component. Well, then you might have that there is some sort of dependency on across strides. Now, when there is dependency across strides, you can be happy about it or not happy about it. So you're happy about it when averaging is not worse than the IID case. You are unhappy about it when averaging is worse than that. For example, if you have a random walk, so maybe one measurement each of the strides, which looks like random walk, then averaging doesn't improve much the variability, because it keeps the variability of the average of observation ruled by random walk. It keeps increasing with n. Just a factor 1/3 is improvement. So there is no advantage. And that's because of positive correlation about consecutive observations. But instead, if there is negative correlation between consecutive observations, then your probability estimate is better, because the negative correlation makes you going back and forth from one side to the other of the mean. So the variability decreases. So it might be that another way to help the variability issue is to investigate a little bit deeper the correlation across strides. So it might be that when you take the average of metabolic cost, once, let's say, you removed all the deterministic components so it doesn't make a mess, and so on, when you average a cost stride, you might have either a positive correlation or a negative correlation of the quantity that you're measuring. For example, the metabolic cost. And if the correlation is negative, then your variability estimate, you want to take advantage of that, because a more detailed evaluation of the variability gives you better estimates, which is naturally there. And so just considering them, not considering the potential correlation across strides, might either inflate or confound some of the effects. So that's what I wanted to say about the first part.
Yeah, absolutely. And it might be worth like exploring maybe like not using consecutive strides and maybe just using strides from like different parts of the trial to kind of like negate that positive correlation.
Good. So awesome, beautiful. So there are a couple of things also that I want to digest first before embarrassing myself with the questions. So I might just send you an email about that. But are there other questions or comments on this? Well, I would say we took away already enough of Dr. Alwan's time. So we just want to thank her one more time for the great presentation and for all the time that she dedicated to this seminar. So also for her great work on the topic. So thank you very much.
Thank you so much. Thank you. Nice to meet you all. Have a good one.